Okay, is everyone able to see my screen? So let me know if in the chat if you're not able to see or if you can't hear me or anything, okay? So evening everyone. Okay, I see some thumbs up, thanks. Okay, so evening everyone, thanks for joining today. So a very short introduction actually about me first. So in case you're wondering what the topics are covered that I'm covering today, these are the following. So I'll be going through a quick revision on cell biology, biomolecules, enzymes. We'll do some practice examples and then there'll be a quiz at the end. Okay, and then I'll drop some tips at the start on how to study biology as well. So a little about me. So I'm a A-levels H2 biology tutor who very recently joined Overmark. So Daryl actually reached out to me. I've been tutoring for about 5 years now. I actually taught GP English at first, and then even though my major in university is life sciences or biology and sociology, but recently I actually started teaching biology more, which is more fitting for my degree. And I recently graduated, so yeah, and which is why I also joined Overmark very recently. So crash courses and one-to-one group tuition, I think those of you who are familiar with Overmark, you all will know more about this. I'll elaborate more at the end of the session, so you can spend more time on revision. Okay, so I'm just going to go straight into it. How do we actually revise or study for H2 biology? Okay, so I think the first thing that that everyone's here should know, and good thing that y'all are here, is that biology, right? I think many students have the misconception that, oh, as long as I memorize, I confirm get A. Okay, there is some truth to that, okay, admittedly, but I think this is what makes many students refuse, or in a way, like, I don't want to say this, but like ego to seek help for biology. Do not be ashamed if you need help for biology, because even though it's memory work, even though at the end of the day you're reading words and chunks of passages and you're just like memorizing them, right? If you do not understand, this can sometimes differentiate between the A and the B students for bio. Okay, so I'm just going to give you 3 golden tips from me that helped me actually get through biology as a student in junior college. So first of all, memory work, very important, okay? Highlighting my maps acronyms, I don't care what, as long as it helps you memorize, okay? You shouldn't care what, oh, later my friends judge me, this is a dumb way of memorizing. No, okay? I myself came up with a lot of weird ways to help me remember. I know some people who listen to music to study, they associate music with like concepts, anything goes, okay? So memory work is key. A tip that I highly recommend would be, for example, drawing figures and all that, okay? So that will really help you condense the extensive text and passages sometimes that you read in your lecture notes. So secondly, your tutorials, revision, all the work that your school provides, unfortunately, as it's very sian to do it sometimes, okay, it's very important to practice for bio, okay? Just like for those who take math and chemistry, physics even, there's a lot of calculations, right? Biology, even though it's just sometimes interpreting figures, writing answers, right, okay, there is the practice element inside it. You need to nail your keywords, okay? Many at times when I was in junior college, I felt super frustrated. I'm not sure if y'all relate. I think some of you might. You know the concept, you write it out, and then you get 0 marks. Why? Because, oh, I use this word instead of the keyword. And it feels very, very, yeah, very sian. Okay, so you need to practice, you need to study, you need to know what keywords you should write when you're answering the questions, okay? And then my last tip will be answering techniques, okay? So biology, just like every other science subject, right? Okay, there's going to be figures, there's going to be essays, okay? What you need to do is you need to be able to interpret the figures, identify key parts of the questions, okay? Sieve out information that the question is actually giving you, okay? And then use the information to your advantage to write to answer the question, okay? And then there's also your paper 3, there's the essay questions, okay? You need to know, okay, given the knowledge that you have, which essay question should I pick? Which question should I do? Okay, so stuff like that. Okay, so without further ado, today I have a lot of slides to go through, okay? So I'll be covering 3 topics: cell bio, biomolecules, and enzymes. I know for biology, actually different schools follow different formats, the way they approach the syllabus. Okay, so I try to cater it to the most logical like progression for biology, and the most I hope most schools syllabus are in common. Okay, so these 3 topics should most often be covered in very early on in the semester. So J1s and J2s, whoever I hear you, this should benefit you all a lot. Okay, so let me share the notes I'm going to use. Okay. Okay, so I hope y'all can see my screen. Okay, so before I start, right? Okay, this is actually what I'm using today is curated notes. Okay, unfortunately, I will not be able to share this with you after the session because these notes are actually meant to be sold as part of the Overmark, the curated notes. They are still very in progress. So what you see I'm using here today is actually just snippets of the curated notes. So actually, if you like the notes, I'm still in the progress of creating them. There'll be a total of 3 booklets by August, which are all released. The first booklet will be out by June. Okay, if you're interested, if you want to supercharge your learning, you can feel free to purchase them on the Overmark website. Okay, so today I'm just using them. What I will give you at the end of the day, at the end of the revision, I'll release on the channel will be a few of the snippets from this snippet. Okay, so it's just very few, but it's just some materials that I don't mind sharing with you guys. Okay, and one more disclaimer before I start. I was a student, I'm student alumni of NYJC, so the notes are very heavily taken from NYJC, but of course I've added in, I've reformatted it, some stuff, I've added in some additional stuff, practice examples, stuff like that. Okay, so just a pre-disclaimer, okay? And you know, yeah, also, yeah, okay? So just to, in case some of y'all are from those JCs and y'all say this one looks a bit familiar, okay? Okay, so for cell biology today, okay, what I want to really drill at is that the key ways of studying not just this topic, but in general biology, okay? So if you notice across biology, every topic, right, there are many comparisons, okay? So cell biology start off with prokaryotes and eukaryotes, okay? Then we go on to plant cells and animal cells, which you will see later, okay? My first key advice to helping you really breeze through cell biology is always make sure you just do comparison when you're doing your study. Do not study, okay, I want to focus on prokaryotes. Oh, I study everything about prokaryotes. Okay, next, eukaryotes. I study everything about eukaryotes, okay? Personally, I don't think that's a good way, a smart way of approaching this topic or most of the general topics in bio. What you should be doing, okay, when you're doing your memorizing is constantly comparing the similarities and differences, okay? Why do I say this? Many biology questions, right, okay, at the end of the day in your exams or your tutorials, right, okay, you'll notice that there are sometimes comparison questions that will pop up. It can be in essay questions, it can also be in the structured questions or even MCQ questions, okay? So, excuse me, it's very important to always do this comparison side-by-side studying, okay? And I will show you what I mean by that later. Okay, so let's start. Okay, prokaryotes, eukaryotic, how to differentiate them? Prokaryotic cells, they lack a true nucleus, lack membrane-bound organelles, the genetic materials. Okay, so you should be thinking of how do you categorize this information? Okay, so for example, genetic organization, genetic material, prokaryotes, how is the DNA organized? Circular DNA molecule, for example, okay? And then we move on to, when you compare it to eukaryotic cells, okay, they have membrane-bound nucleus, membrane-bound organelles, okay? How are the DNA organized? DNA molecules associated with histone proteins, okay, to form chromatin, okay? So one more thing that I think is really important, for example, would be a hierarchy, hierarchization. I don't know how that's the word, but to create a hierarchy. So what do I mean? Okay, if I can draw your attention to, okay, yeah, over here. So if you notice this, examples of all eukaryotes, okay, so you can see prokaryotic, eukaryotic cells, okay, imagine there's two categories, so prokaryotic and then eukaryotic, okay, and then you further branch it out. So eukaryotic, what are eukaryotes? So for example, we have plants, okay, so you can write plant cells. Okay, I'm not going to write the whole thing. Okay, you have animals, okay, animals, okay, and then fungi, and so forth. Okay, prokaryotic cells, what do you have? Okay, we have bacteria. Okay, just for example. Okay, so this is how you should be approaching your learning when you're rememorizing and revising all these topics. Okay, one important thing is because most often when you study biology, once you move on to the next topic, right? Okay, you may tend to, okay, I don't need to care already, I finished. And then that knowledge slowly starts to become very muzzled, it fades away. Okay. This is a bad habit. I did it because I was lazy as a student as well. Okay. What you should do is, I would think that as you approach new topics, right? Okay, always spend a few minutes beforehand. I mean, when you start the new chapter, okay, to revise the old chapters, okay? Because at the end of the day, right, okay, much of the topics in biology, it's very linked together. Okay, why did they start off with cell biology first? Okay, because if you see that cell biology, right, it actually links to your DNA and genomics. Okay, DNA and genomics links to cell division. Okay, cell biology also links to your extension topic of infectious diseases. It also links to respiration and photosynthesis. So if you notice all the branches out, okay, so if you forget your cell biology, right, okay, then you may find, wow, your later topic is a bit GG lah. Okay, so always try to spend some time to revise all your older topics. Okay, it doesn't mean you need to reread everything. Okay, but more of just spend 5 to 10 minutes, just quickly skim through everything. Okay, okay, so let's move on. Okay, now this is what I mean by you guys should be doing comparisons when you study. Okay, so if you notice here, okay, let me press my pen. Okay, so notice this table, what I've listed here, right? Okay, similarities and differences. When you do comparison, right? Okay, always try to just whatever you can think of, whatever similarities, just plop it into a table. Whatever differences, just plop it into the table as well. Okay, so most schools, I think your lecture notes, they may provide this for you. Okay, this is why I think it's a very effective technique of memorizing content because you save a lot of time. Why do I say this? Okay, so common stuff, similarities, right? Okay, it just cuts down the things you need to memorize. Okay, if you rather than you think of prokaryotic and eukaryotic separately, right? You're memorizing this for, let's say, prokaryotes. Okay, and then let's say you're done. Then for eukaryotes, what do you need to memorize? This again. Okay, so you can see there's a bit of double work when it comes to the similarities. Okay, and then like, because for some people I know you memorize, you're like, okay, prokaryotes, they have 6 factors. Okay, then you start to name the 6 factors. And then eukaryotic, you're like, okay, there's 7 factors, then you start to name the 7 factors. Okay, notice there's a lot of double work done. Okay, rather what you can do instead, okay, 4 similarities, okay, and then the differences are on what comparison, what level of comparison, okay? So for example, the presence of nucleus, okay? As we've covered briefly just now, prokaryotic cells, no true nucleus, eukaryotic cells have a true nucleus, okay? So this is what I mean by you should be doing a comparison in terms of a revision, okay? Okay, so moving on. Okay, I know I'm going a little fast. Okay, there's actually a lot of slides as you can see. So the point of today is not to super like content dump you guys, but more to provide some revision skills and of course to recap certain topics that I think it's important that may come up for your mid-years. Okay. Okay, so besides comparison question, okay, this one is another thing that I would want to point out. Okay, so in your lecture notes, right, sometimes you may see certain areas that that you feel like, oh, this one confirm won't test one now. Okay, always make sure to check properly, read unless your teacher explicitly mentioned that this is for your information. Okay, it is possible that it can be tested for your exams, A levels or whatnot. Okay, so take for example cell component, the size of your cell organelles and all that. Okay, this came out for one of the A levels. I forgot which year, but MCQ. Okay, so stuff like that. Okay, you may see this is in your lecture notes. It may just show like that, and then you're like, oh, okay lah, just skip it over. Okay, not necessarily. Do not do that because your school, right? Beyond your school, exams and questions, right? They can take this information and test it in a various number of ways. Okay, so they may not ask you for the exact diameter. What they can ask you instead is perhaps they will list a few organelles and ask you to rank them in increasing order or decreasing size. So that could be an MCQ question. Or if your school really, really wants to torture you guys, okay, they can make you do calculations. So they can give you the microscopic level, they can ask you to estimate the size, or why is this even important? Okay, they can show you electron micrograph, okay, and then they can show you this very, you know how bio, this is meme, they all the figures are always very distorted, very hard to see. Okay, so sometimes they will provide this, for example, this you may see this in the electron micrographs. So 5 micrometers. Okay, so this is just an estimation. This is to show you the measurement of the specification, the size, okay, in the electron micrograph. Okay, so how can this help you? Okay, so sometimes, let's say this is the picture lah. It's like a cell, and then this is the measurement. So they show you this measurement is for good reason. Okay, so allow you to measure and determine the size of the organelles. Okay, why is that important? Because they may give you 2 very similar looking organelles. For example, your chloroplast and mitochondria. Okay, and then they may expect you to identify. Okay, so if you, let's say you didn't memorize this table, or yeah, we never study really well cell biology, okay? But if you roughly remember, okay, which one is larger, chloroplast is larger or mitochondria is larger, okay, you can tap onto that information to help you. So sometimes diagrams, right, okay, they may seem like they're dumping a lot of unnecessary information, okay, but they're actually, most of the information that actually can be used by you to help answer the question, okay? They help you refresh your memory or whatever, okay? So important point I wanted to raise here in case some of you may think, oh, this is not important, okay? So yeah, okay, moving on. Okay, so as I mentioned, plant cells, animal cells earlier on, they're both eukaryotic in nature, okay? How to memorize this part? Very simple. Besides the common organelles that they have, for example, your cytoplasm, your nucleus, okay, your vacuole, for example, okay, what is important to understand? Differentiating factors. So for animal cells, we have lysosomes, centrioles, flagella. Okay, two that are present in animal but not necessarily in plant cells. Okay, so yes, unfortunately, at the end of the day, there's still a lot of memory work. You still need to memorize all these different. Okay, so I've included here the ultra structure. You don't really need to go and memorize like everything in so detailed. Okay, because some of the organelles listed here, they are actually included in processes that we'll look into later. Okay, and then along the way you'll memorize. So what I mean is you don't go and memorize like blindly, like just, oh, okay, animal cell, there is secretory vesicle, there's Golgi vesicle, there's Golgi body. No, okay? Memorize certain parts as part of the process. So what do I mean? Golgi vesicles and secretory vesicles, okay? You memorize it as part of the endomembrane system, which I will explain later. Okay, so memorize the things that are constant. So for example, your cytoplasm, your nucleus, these are just some examples, some key examples that you really need to understand for in the structure of cells. Okay, so that is it for animal cells, and same thing for plant cells. Okay, what are the differences? Over here, the keywords. Okay, so we have chloroplast, a large central vacuole, tonoplast, cell wall, plasmodesmata. Okay, so the thing about biology is that it often builds onto your knowledge from your previous year. So secondary knowledge, your cell bio, you learn parts and bits, right? Okay, and now JC, you learn a lot more. Okay, so the thing about bio, okay, preface here, okay, don't feel demoralized when you get confused or when you feel that it's so overwhelming, okay? Trust me, I'm at university, I take biology, okay, I'm still learning. Okay, I graduated, but I can tell you that I'm still confused at a lot of the stuff. Biology is a field that is constantly evolving. I can tell you straight up that in JC biology, right, okay, they simplify a lot of things, okay? So take it one step at a time. Sometimes when you're studying, right, okay, questions may pop up in your head that may make you even more confused, okay? Do not be afraid to approach your tutors, your teachers for answers, okay? And to clarify all these doubts. Okay, most of the time, sometimes you may not be satisfied with the answer. It's because at your level, okay, there are certain information that you need not necessarily know. Okay, so it's fine. Okay, as long as you understand the processes, I think it's a good to go for your A levels. Okay, I think one trap you should not fall into is to, if you feel like you're just memorizing for the sake of memorizing, someone asks you a question, you can recite it fully. Let's say I can recite this full page fully, but you don't understand anything. Okay, that's a very dangerous, it gives you a false sense of confidence, because when you go into examinations, right, okay, there are recall questions, yes, you will ace those, but when it comes to application questions, analysis questions, you may tend to suffer a bit more. Okay, so yeah, okay, always make sure you try and understand the key concepts as well, okay? At this point, if there's any questions, feel free to type in the chat. I will look at them at the end of the session or as and when, okay? Okay, so now, okay, so I thought it would be nice to do a very quick exercise here, okay? Don't worry, okay? I'm not gonna make anyone unmute themselves. This is more of revision for your sake, okay? So what I mean by the comparison table, okay, I've done it up here as well. There's the field one later, okay? We'll look at it later. So same thing here, comparison between plant and animal cells, okay? So maybe I can get some okay, let's do the similarities together, okay? So maybe you can type some similarities in the chat. So okay, I'm gonna open the chat. Okay, so can I get some similarities in the chat? So as many as you guys can think of. Okay, maybe I will type. Yeah, so to start off, right, okay, maybe one example, very straightforward one, both have cytoplasm, okay? Okay, so I'm typing in very layman's language now, okay? So you should not be answering questions like this in point form or that unless specifically stated, so you should always answer in full sentences, okay? So any answers? Okay, good. I see one membrane-bound organelles. Very good. Okay, you're building on what was mentioned initially. Eukaryotic cells, plant and animal cells are both eukaryotic cells, so their organelles are both membrane-bound. Very good. Okay, so I'll just put. Okay, good. So very simple. Cytoplasm, membrane-bound organelles. Okay, what other similarities anyone can think of? Okay, if you're shy, you can direct message to me on Zoom as well. Don't have to send on the or chat. Okay. Okay. I'll give another one, very simple one, if you can think of. Okay, when you think of cells, okay, the first thing that you should always think of also is your genetic material. How is genetic material organized? Okay, so for both plant and animal cells, what is it? Okay, immediately you should think of DNA. Okay, so their genetic material is in the form of DNA molecules. Okay, and where is this information actually coming from? It's actually coming from earlier mentioned the eukaryotic cells as well. Okay, eukaryotic, prokaryotic, remember? So prokaryotic is circular DNA. Okay, but for plant animals, it's DNA molecules in the form of, so for example, chromatin, histone proteins. Okay, so okay, any more? Okay, both have mitochondria. Very good. Okay, both have mitochondria. Okay, good. So good answer here, both have mitochondria. So sometimes some students may get tricked. They often study mitochondria and chloroplasts together. Okay, so be careful, very good that both have mitochondria, but remember that chloroplast is exclusively for plant cells. Okay, and I think it's also because it's in the table over here. Okay, thanks for your participation. Okay, so we'll move on. And you can see the field table here. Okay, so don't worry. I think in the notes that I'm going to send afterwards, this will be there. Okay, so for your own revision, very nicely condensed. So here, notice that cytoplasm, membrane-bound organelles, yes, cell surface membrane. Okay, DNA is the genetic materials. Okay, nucleus. So you can see this. Sometimes when you're studying, you may feel like, oh, okay, this is very straightforward. No, okay. Biology, always make sure you list up everything. Okay, info dump everything, okay, to make sure in your revision, in your learning, okay, so that when you recall the facts, right, okay, you make sure you cover every aspect of the whatever topic you're revising, okay. So moving on to the what's true nucleus. Okay, good question. Okay, so true. Okay, so at your level, right, okay, so you just need to know that for the nucleus itself, right, okay, as we'll go into the structure later, there's like the nucleus, nucleolus, okay, so understand that for plant and animal cells, right, okay, there is just this presence of this nucleus. For trying to think how to not info dump. Okay, so for further on, okay, specific cell types, right, okay, they may lack this true nucleus. So what do I mean by that? Sometimes their DNA need not necessarily be organized into like the nucleus in a cell, okay, which is surrounded by like a membrane. So you'll know later, you'll learn that like true nucleus is like the nuclear envelope and all that, okay. So for certain organelles, for certain organisms or certain cells, okay, their nucleus need not necessarily be wrapped in the nuclear envelope. Okay, so sometimes this is more dynamic in nature. Okay, so put it simply, true nucleus means it's membrane bound, okay, by the nuclear envelope, okay, which we will see later. Okay, so I hope that clarifies. Okay, so moving on, these are very points that are based off the plant cells, if you notice, where the comparison can be drawn. So if you see plant cells, right, okay, we have cell walls, okay, we have chloroplasts, okay. So the differences that were mentioned just now over here, you can see here, okay, and here, okay, these just add on to the table. to differentiate your plant and animal cells. Okay, so, okay, clear? Okay, so make sure that, once again, I can't mention this enough, when you're studying, make sure you do your comparisons. Okay? Okay, okay, so now moving on to your, okay, so for the next part, right, okay, I will be roughly going through the organelles and their functions, okay, but I'll be focusing on certain processes which I think is important for your cell biology, okay, understanding. Okay, so as I mentioned just now, the good question about the true nucleus, okay, so for nucleuses in plant and animal cells, right, okay, they are bound by a nuclear envelope. So, excuse me. So the double membrane, okay, which you will learn later about the cell surface membrane, a phospholipid bilayer, okay, surrounds the nucleus. Okay, so the nucleus is more often referred to as like the area where genetic information is stored, okay, like the control center of the cell, okay, or if you want to do an analogy, the cell, it's like the cell's brain, okay. So we have nuclear pores, okay, because we understand that subsequently you'll learn that transcription, translation, right? Okay, it's a very dynamic process. Okay, it occurs in different parts of the cell. So when these processes occur in different parts of the cell, right? Okay, you're having materials that are created and needed for these processes to move in and out of the cell. Correct? I mean in and out of different organelles. Okay, so we have these nuclear pores which allow for the movement of these molecules. Okay. Okay, so what I think it's important to mention here will be for your chromatin. Okay, so chromatin, which I mentioned just now, this is fully explored in one of the subsequent topics of DNA and genomics, okay? But at the base, what you should know here is that DNA is the genetic material of the cells, okay? It's wound around histone proteins to form chromatin, okay? So yes, there's euchromatin and heterochromatin. So euchromatin is the less condensed form, and then they are frequently transcribed. Heterochromatin is more highly condensed. So for me, how I memorize this is, for example, heterochromatin, H, highly condensed. So, and then if heterochromatin is the highly condensed one, it means euchromatin is the less condensed one. Okay, so I'm going to tap into a bit of, I'm not covering DNA genomics today, but I think what it's good to help you guys with, okay? Some students are often confused. Why is it that it's less condensed means frequently transcribed, okay? So when you're talking about condensed form of the DNA, right, okay? Think about it as transcription as, let's say you want to read the DNA polymerase, right, okay? No, not polymerase, okay? So it wants to read the genetic information to transcribe it into mRNA, correct? So it wants to read the DNA, which is like a book, to transcribe it into mRNA, let's say another book, okay? So if it's less condensed, think about a book that it's very easily open, very, very loose. So I can easily open up the book and then I can read the content to transcribe it, correct? Okay, so that's why it's more frequently transcribed. But if the book is so loose, I can just open it and look at all the material and transcribe it, okay? But think about highly condensed chromatin. So think about the same book, but now it's bound, it's very tight, okay? I can't really open it, I can't flip it as well. Okay, let's say if I want to open it, I really need to use all my strength to open it, to read the stuff inside. Okay, and if that is the case, it's going to take a lot more effort to transcribe it into mRNA or the other book, correct? Okay, so this is why highly condensed chromatin is less often transcribed. Okay, generally not transcribed. Okay, because they are very difficult to access the materials inside. Okay, you really need to go and open up the DNA, okay, to go and access the genetic information to transcribe it, which is why it's generally not transcribed, because it's already so compact. Okay, nature doesn't make things hard for itself. Okay, so I hope that analogy helps you to better understand for your DNA and genomics. Okay. Okay, so moving on, okay, another key part that you need to understand for the nucleus would be the nucleolus, okay, which is where your ribosomes come into play. Okay, so remember just now I said about using whatever it takes to get your memory work done. So over here, this figure, notice that all the information in the text above, right, condensed into here. Okay, this really helps with your understanding of the revision. Okay, very simply put, okay, if I can zoom it in, okay, here you can see that nucleus, nucleolus, okay, and then the white space outside is your cytoplasm. Okay, so Nucleolus, as I mentioned earlier, it's part of your ribosome biogenesis. Okay, it's very important that you follow these arrows clearly. Okay, so you can see that ribosomes, right, what are they made out of? Okay, ribosomal proteins and your rRNA. Okay, and then that gives your small and large subunit. Okay, so you can see that it's very delicate. The ribosomal proteins, okay, they have to go in to the nucleus, into the nucleolus, okay, to assemble with the rRNA, okay, to give rise to your small and large ribosomal subunit, okay, which are then assembled in the cytoplasm to form your cytoplasm, to form your ribosome, sorry. Okay, so you can see by the arrows. Okay, so one question that may pop up, how do you think the ribosomal proteins enter the nucleus? Okay, what did I cover just now? Okay, the presence of nuclear pores, which it's not illustrated in the diagram. Okay, but so these nuclear pores, these ribosomal proteins are able to move through the nuclear pores into the nucleolus, which is their site of site of assembly. Okay, now key point here. Okay, so when key point here, okay, keywords. What do I mean by keywords? Okay, so I recall this question. Okay, they're asking me to describe about how are ribosomes formed, okay, or how are ribosomes produced in the cell. Okay, I remember clearly my answer. I wrote rRNA and ribosomal proteins combine in the nucleolus, okay, to form the large and small ribosomal subunits, which then form the ribosomes in the cytoplasm. 0 marks. Okay, why? I was very shocked as a student because I thought I nailed the concept. It's based off this. Okay, why? Keyword that was missing, assembled. Okay, so the thing about biology, right? Okay, you need to make sure is that certain keywords, certain of the verbs, right? Okay, like formed. Okay, sorry, handwriting very ugly. Okay, formed, and then we also have. Okay, I'm just gonna type assembled. Okay, so stuff like that. These keywords to the layman, right? Okay, it may not mean like, oh, there's not much difference. They're formed or they're assembled, or it's the same thing. At the end of the day, got ribosome come up. Okay, for biology, if it's highlighted as a keyword, right, more often than not, you really need to use the exact word, because if not, even though you got the full concept, right, if you got the full concept correct, okay, you may get 0. Like that's the truth. Okay, you may get 0 because you're not hitting the keywords. Okay, so even if you got the concept correct, okay, if you do not hit the keywords, for example, over here, the nucleolus is the site of rRNA synthesis. Ribosomal proteins are imported from the cytoplasm and are assembled with the rRNA into the large and small ribosomal subunits. Okay, if this keyword is missing, you can get 0. Okay, so harsh reality, but because certain keywords have more scientific, like there's this sciencey words lah, okay, that you need to include in your answers. If not, they don't really count. They don't really make sense from a scientific point of view. Okay. Okay. So I hope this is clear. If any questions, please feel free to mention or stop me. Okay. Another confusing part. Okay. So the ribosome itself. So always remember, notice so far that for every organelle that I'm going through, right? Okay, I'm doing structure. What is the structure? What does it look like? What is it made of? Structure, and then structure in relation to the function. So remember for the nucleus, I mentioned that, okay, what is the structure? Okay, it's a membrane bound, it's where the DNA, the genetic material is stored. What is the function of the nucleus? Okay, the nucleus is the assembly site, is where the rRNA and the ribosomal proteins are assembled, so forth. It's where genetic material is transcribed, so forth. Okay, always relate structure to your function. Okay, so ribosomes, they are small, they are non-membrane bound organelles. So this is a key word that you should take note of. They are non-membrane bound, okay? And they consist of a large and small subunit. Each subunit is further made up of of rRNA and ribosomal proteins combined together, okay? So they can exist as free ribosomes floating around the cytoplasm, or they are specifically attached to the RER, which I'll explain later, okay? So key thing to note, another differentiating factor, which I think was listed in the table just now, ribosomes in prokaryotes are known as 70S ribosome, eukaryotes 80S ribosomes. Okay, another differentiating factor. Make sure to, when you're studying this part, you can jog your memory a bit. Okay. Okay, some of you might ask what is the like 70S, 80S, what is the S? Okay, not necessary to know, but if you are curious, S actually refers to it's spelled as, I don't really know how to pronounce it. Separate units. Okay, so you can see separate units. I don't know how to pronounce, sorry. Okay, but this unit actually refers to the sedimentation coefficient. So it's more of how fast they sediment. So ATS for the eukaryotic ribosomes just means they sediment faster. Okay, so you don't really need to know this at this level. Okay, so one thing to notice here, the numbers. Okay, the sedimentation coefficient is not one-to-one addition to get the number. Okay, so this is where some students might be confused because they might think, oh, okay, 50S plus 30S should give me 80S. What? Okay, no. Okay, how I remember this is that for prokaryotes, okay, at the end of the day, the number is smaller. So even though this totals up to 80S, okay, the prokaryotic ribosome is 70S, correct? Oops. Yeah, 70S, okay, it's 70. Okay, and then the eukaryotic ribosome, even though this totals up to 100S, okay, the eukaryotic ribosome is 80S. So at the end of the day, eukaryotic is still larger, regardless of what. Okay, so just remember this. Okay, you don't really need to go and memorize all these small details. Okay, it's not necessary. Okay, so function of ribosome, very key part. The ribosome is a very key organelle. Okay, it plays a role in protein synthesis. Okay, so proteins are very important, which I'll cover in the biomolecules later. Okay, so I'm gonna speed up a bit because we're still at only cell biology. Okay, so the key parts of cell biology that I think you guys should really understand will be, besides each organelle and its specific functions, the key parts you need to study are the main organs, the main organelles such as the nucleus, the cytoplasm, endomembrane system, I'm typing it out, endomembrane system, and the protein synthesis. Okay, so over here. So these four are the main that you should really understand. Oh, sorry, actually there's one more. Mitochondria and chloroplast. Okay, so these five. Okay, I know this is like, you're just telling me to study everything. Okay, but what I mean is you understand the cell biology in these different sections. Okay, so protein synthesis, what is just basically the ribosome, what I mentioned just now. Okay, now the endomembrane system is what I'm going to cover now very quickly. Okay, so I'm not going to go too much into the specific structure and functions. Again, this you can do on your own. Okay, what I want to mention is, for example, the entire system. So these, whatever is above, right, you can, if you buy the curator notes, you can revise that. It's all the functions very condensed nicely for you. Okay, what I really want to go through would be, for example, the process. Okay, and for that, we need to look at the Golgi, the functions of the Golgi. Okay, many students tend to forget that protein synthesis is a very dynamic process. Okay, it's not just, oh, I synthesize the protein, done. Okay, we need to think about the protein shape, the function, how the protein, where's the protein going to do its function? Is it function in the cell or outside of the cell? Because remember, your cell is just a piece of you, okay? It's very dynamic. It interacts with other cells. It plays a role in the tissue, in the organ. They're interacting with other cells. So sometimes they need to secrete the protein to do into the external environment to carry out its functions, okay? The Golgi's main role is on chemical modification. And when we mention chemical modification in cell biology, automatically you should be thinking about glycosylation and phosphorylation, not glycolysis, sorry. Okay, glycosylation and phosphorylation. These are chemical modifications. Okay, glyco. As you go through biology, you should know that glyco refers to sugars, carbohydrates. Okay, so oligosaccharides, your glycoproteins, glycolipids. Okay, depending on what they add the sugar, the carbohydrate group to. Most often, if it's protein synthesis, we're talking about formation of glycoproteins. Okay, when you add carbohydrates to the proteins. Okay, and then we have phosphorylation as well, which is where you add your phosphate groups. Okay, so it's we talk about Golgi. There's a lot of packaging, modification, temporary storage, and stuff like that. Okay, okay. So I'm gonna skip through all this because yeah, these are just your content that you should know. Okay, mitochondria and chloroplast. Okay, very similar, same thing. Once again, look at this comparison table again, which you should really follow. Okay, I think it's really helpful to do your comparisons. Okay, double membrane, single circular DNA, binary, can divide by binary fission, ribosome. So if you notice that, for those who are a bit more keen-eyed, okay, you may notice that mitochondria and chloroplasts, they tend to resemble prokaryotic cells, okay, which is where we talk about the endosymbiont theory, okay. So I'm not sure if you guys are familiar with this theory. It's covered, so it's important to know this. There is this biological theory that our mitochondria and chloroplasts are actually derived, like they're actually used to be cells on their own, but they form this endosymbiotic relationship with our, that's why in our eukaryotic cells nowadays, okay? And this is because many similarities exist between the mitochondria and chloroplasts and other prokaryotic cells. So if they ask, questions can be testing you, for example, what are the similarities between chloroplasts, mitochondria, and prokaryotes? And then you as a student, you may be shocked because you're like thinking, huh, chloroplasts and mitochondria, they are organelles, they are not cells, okay? Which is why this is what I mean by certain parts of the content, they can twist it to ask you application questions, okay? So the key facts here are mentioned earlier. Okay, 70S ribosome, divide by binary fission, circular DNA, okay? Okay, so unfortunately, I don't think I have time to go through the cell membranes. I'm going to skip all this. And we're moving on to the next chapter, which is on biomolecules. Okay. So any questions, please feel free to just send into the chat as well. I'll try to answer all of them at the end. Okay. Moving on, biomolecules very quickly. Off the top of the head, three things we need to revise: carbohydrates, lipids, proteins. Okay. Nucleic acids will cover. I'm not covering today, but it's DNA and genomics, so separate out. Okay. So the three major biomolecules that we need to cover today: carbs, lipids, proteins. Okay, so we start with carbs. Okay, very simple. The thing about biomolecules, you always need to understand the different levels of organization. So what do I mean? For carbs, we have monosaccharide, disaccharide, polysaccharide. Okay, the key is in the name itself. Mono means one, di means two, poly means a lot. So monosaccharide, one sugar. Okay, so what do we have? Glucose. Okay, the key molecule that you really need to really understand for carbs is glucose. Okay, the key part to memorize here, you differentiate your alpha glucose and your beta glucose. Okay, so for me, how I tend to memorize this is, for example, alpha glucose. Beta glucose, right? Okay, so the difference is in this carbon over here, correct? Okay, so for me, I remember that, okay, beta glucose, B, bottom. Okay, but then I have to tell myself it's very confusing for some of you. That's why what I say is like it's really up to you yourself. Okay, so for me, I would say beta glucose, carbon 1, bottom. Okay, but it's the opposite. So the OH is on top, the hydroxyl group is on top. Or for some people, how they memorize is, okay, for beta glucose, the hydrogen on carbon 1 is on the bottom. So by that extension, okay, the hydroxyl group is on top. Okay, and then if beta glucose is this structure, alpha glucose will be the opposite. Okay, so the OH should be at the bottom, the hydrogen group will be on top for carbon 1. Okay, so really up to you how you memorize. I'm just sharing tips on how I memorize. Okay, so important to note for monosaccharide glucose, alpha glucose and beta glucose is how the carbon 1, where is the hydroxyl group? Okay, where is it projecting? Okay, and what is the main thing about glucose is that it is the substrate for ATP production, which is covered in respiration subsequently. Okay, so I'm not going to go through fructose and galactose. Okay, next. Now, the key thing to know about disaccharides would be how are monosaccharides combined to form disaccharides. Okay, so we are moving on. See, it's very sequential: monosaccharide, disaccharide, and then polysaccharide. Okay, so when it comes to disaccharides, okay, what is the key thing you need to understand? Hydrolysis and condensation. Okay, so how are two monosaccharides combined to form a disaccharide? Condensation, bringing together, okay, with the removal of one water molecule. Okay, so very simple. How is the glycosidic bond named? Okay, depending on the first molecule. Okay, so let's say here, okay, we have an alpha glucose. Okay, it's combining with, don't care what, it's combining with a second glucose, right? Okay, but with alpha glucose, so this is alpha. Okay, next thing, where is the bond being formed? Okay, what are the two carbons, which are the two carbons that are interacting? Carbon 1, okay, so alpha 1, and this is 1, 2, 3, 4. 4. So alpha 1, 4, glycosidic bond. Okay, this is how you really process. So why am I actually slowing down to go through the how to determine where is the bond form? Okay, it's because sometimes they may give you very complicated diagram. This is just one disaccharide here. Sometimes they may show you a very big figure, and then they may ask you to pick out where are the bonds being formed and what is the bond. Identify the bond being formed. Okay. Okay. So, and then hydrolysis. Okay, it's the opposite. If condensation gives you one water molecule, okay, hydrolysis requires the addition of one molecule. Okay, so for those of you who are better in chemistry, you should know this. Okay, so addition and removal of the molecule. Okay, it's a very balanced process. Yeah. Okay, now I'm gonna skim through. This is actually included in the slides after, so don't worry about jotting everything down. You can go through this later. When we talk about biomolecules, right, always remember the different organizations. So here, right, okay, what I'm actually showing here is your polysaccharides, okay, because it's the polysaccharide that is the one that is carrying out your functions, okay. Your monosaccharide and disaccharide is more of teaching you the conceptual, equipping you the conceptual tools to understand how the polysaccharides are built up. Okay, so we have starch, glycogen, and cellulose. Okay, how to remember this? Separate it out into what are the functions. Okay, so we have starch and glycogen, which are more of for energy storage. Okay, and if you remember, glycogen is for animals. Okay, and then starch is for plants. Okay, so how to remember? For me, I just remember like potatoes, vegetable. So there are a lot of starch. Okay, potatoes, yeah. Okay, I just tell myself, okay, that's a vegetable. Okay, so if starch is vegetable, if starch is the storage molecule for vegetables, plants, okay, we have glycogen for animals. Okay, cellulose is more of for structural support. Okay, so take note of the structure to function. So I think here, when you guys get the notes, what you should always study and focus on is here. Okay, you should understand the properties in relation to the function. Okay, because starch and glycogen are large and they are compact. Okay, means you can store a lot of stuff and they won't take up a lot of space because they're so compact, which makes them good for storage molecules. Okay, but if we're talking about structural support structure, you want rigid, you want strong. Okay, so which is why we have cellulose. Okay, they are very strong polysaccharides. Okay, moving on. Now lipids, I'm not sure. Okay, lipids very quickly because I'm running out of time. Okay, so for lipids, same thing. Always make sure you study properties in relation to function. Okay, so when we talk about triglycerides, your typical fats, okay, fatty acids, okay, where they are large, non-polar, insoluble in water. These are the properties, but how does this translate to their functions? Okay, we have energy storage. Think about blubbery animals, so like whales and seals, right? They have a lot of fats. What does that mean? They're living in what cold, like North Pole, all there, right? So they have a lot of, and they need a lot of energy to store inside. Okay, when they eat, food is not always readily available. Okay, they have heat insulation because it's so cold. They have buoyancy, keep buoyancy, protection. Okay, because fats, they help you cushion impact. Okay, phospholipid, this is part of your cell membrane, which unfortunately I cannot cover. And then cholesterol is embedded in your cellular membrane, so helps to regulate membrane fluidity. Okay, now what I want to spend a bit more time is your proteins. Okay, so proteins, right? A key concept that you really need to understand would be your primary, secondary, tertiary, and quaternary structure. Okay, so many students tend to get confused between tertiary and quaternary structure. Okay, so tertiary structure, just remember always that it's the unique 3D conformation. Okay, so which is where your 4 bonds should come in. Okay, so here only mentioned 3: hydrogen, ionic, hydrophobic interactions. There's actually one more, your disulfide bonds, which is not always present, but it's possible for it to be present. Okay, disulfide bond, which is a form of covalent bonding. Okay, so at each level, right, your primary, secondary, tertiary, and quaternary, okay, there are specific bonds that you need to understand. So that is the important part. Okay, primary structure, we have your peptide bonds, which is a form of covalent bonding between amino acids. Secondary structure, we have your hydrogen bonding, okay, between your peptide bonds. So you can see it's a very sequential manner again. And then tertiary and quaternary is actually the same thing, but tertiary structure is your within the same protein molecule. Okay, so don't get confused. Even though their bond types are the same, hydrogen, ionic, hydrophobic, and disulfide bonds, quaternary structure, these bonds exist across different polypeptides. Okay, so I can have two proteins or two polypeptides. Polypeptides and proteins are the same thing. Remember, polypeptide means many peptides, which is a protein. Okay, so I can have two proteins, two polypeptide chains. Okay, they may overlap with each other and fall with each other. This is the quaternary structure because it involves two polypeptide chains. Okay, so I hope that is clear. Okay, so and then for proteins, we're talking about the big molecules. What are their functions? We have hemoglobin and collagen. So hemoglobin, as you guys know, it's more of a, from diagrams you should see, it's more of a globular shape. So we can classify it as under a globular protein. Collagen is more of fibrous. Okay, so remember just now your cellulose? Don't confuse cellulose and collagen. Cellulose is carbohydrates, collagen is protein, but both are meant to provide structural support. So collagen is more of a fibrous protein, structural support. Okay, you can see same properties. Insoluble, high tensile strength, rigid, compact. Okay, hemoglobin is more of a special case. It's a globular protein. It's meant to transport oxygen. So it has to be soluble because it's a blood protein. Compact. Imagine if it's very big. Okay, you don't have much hemoglobin. It's difficult to transport oxygen in your body. Okay, so and then its function is laid out nicely over here. Okay, so okay, I know that is very fast, but okay, we are moving on to our last last topic before we move on to the practice questions. Okay, so enzymes. Okay, very quickly, enzymes. Four main things you need to know. Okay, so the mode of action. The key characteristics of enzymes: their mode of action, factors affecting enzyme, and then the inhibition. So inhibition, like your competitive inhibitor, non-competitive inhibitor, okay, which unfortunately I will not cover today. So for enzymes, right, the key things you need to know are its specificity, need in small amounts, what is their function, they remain chemically unchanged, they are like a catalyst, okay, the keyword, people always refer to enzymes as catalysts, okay, so they are affected by what factors? Substrate concentration, enzyme concentration, temperature, and pH. So 4 factors, okay? When you're revising, tell yourself how many factors are affecting enzymes? 4. Okay, so these are some parts that you need to remember, like in terms of numbers, okay, to help you aid with your memory work. Okay, so moving on, okay, the mode of action, which is what I really want to cover. Enzymes, right? Okay, do not forget at the end of the day, they are proteins. So remember just now at the start of the lesson, I mentioned about how topics are related to each other. Protein, what we just covered, their functions, they can be functional proteins, right? Like your hemoglobin and your collagen, right? Okay. At which level do they carry out the activity? Okay. The tertiary level. Because the tertiary level is where their unique 3D conformation is formed. Okay. And then that is where, so they fall into a unique conformation, right? And that is where for enzymes specifically, your active site will appear because they fold very nicely. They over all the amino acids folded, okay? And then there is it folds into this shape, okay? And then that is where your active site is. Question: is it possible for a quaternary structure to have the active site to be found in the quaternary structure as well? Yes. Okay, because certain enzymes, right? Okay, they need additional proteins to, let's say, combine together to form the quaternary structure, and then the active site is formed. Okay, so that's a key point that I want to raise, okay, in case it ever comes up. Okay, so with that, right, given that the enzyme structure is so unique and specific, what does that mean? If I fold until the groove is the active site is very specific, right, means it can only take a specific type of substrate, which is what I mean by enzyme specificity. Okay, so it must be complementary to the substrate. Shape, size, charge, orientation, SSOC acronym. Okay, important to memorize. Okay, now moving on to the how enzymes carry out their activity. Okay, what we know as the enzyme substrate complex. So once you form the thing, right, it's complexed together. It forms the enzyme substrate complex, which is where they'll do the magic will happen. Oh, okay, they'll carry out the activity. And then the enzyme, remember, it remains chemically unchanged, and then the products will be released. Okay, so I hope this is clear. Next, the substrates are held in active site by weak bonds such as hydrogen bonds, ionic bonds, and hydrophobic interactions. Okay, so yes, they say weak bonds here, but there's differing levels now. Okay, we know that hydrophobic interactions, hydrogen bonds, ionic bonds, and then disulfide bonds. So question you may ask here, why are substrates not held by disulfide bonds? Okay, answer, disulfide bonds are, as I mentioned, covalent bonds. Covalent bonds require super high amounts of energy to break. And we do not want the enzyme substrate complex to be so tightly bound together. If not, it's not efficient. Okay, imagine if every time you want to form a product, it needs to be tightly bound and then it needs so much energy to release. The whole point of enzyme is to lower the activation energy. So this is unnecessary. Okay, so which is why it's held by weak bonds. Okay, okay. So the two hypotheses, unfortunately, I will skip through it, but you just make sure you remember the induced fit and the lock and key hypothesis. Okay. Now, what I want to cover, okay, is actually the factors. Okay. So as I mentioned just now, substrate, the four factors: substrate concentration, enzyme concentration, temperature, and pH. Okay. So substrate concentration, everything you should study, right? Okay, you should study substrate concentration and enzyme concentration in relation. Okay, study these two together. Now, for enzymes, right? I think for me personally, how I like to understand this is I always try to rationalize it out or by drawing it out myself. Okay, so you can, for example, draw. Okay, let's say I have an enzyme. Okay, in my solution lah, I only have two enzymes. Okay, but I got a lot of substrate. Okay, oh wait, sorry, this is the next one. Sorry. Okay, so if I have a lot of enzyme, okay, but I only have two substrate, so you can see that the active sites are not all occupied. Okay, which is why the rate of reaction is very slow, because I still have a lot more. Let's say I got a lot of enzyme, but I only have a few substrate. So the rate of reaction is very slow because this one, but here these three enzymes are unused. So the moment I add in more enzymes, so long as there are more empty active sites, okay, you can see that the rate of reaction will immediately go up. Okay, which helps to explain the curve because active sites are not all occupied. But the moment I add in 1:1, so you can see it's a very linear increase. At low substrate concentration, there is a linear or proportional increase in the rate of reaction. But the moment I add in too much substrate, what happens? The rate will slow down because you can start to see if I add in more, this is still being occupied. This is still being occupied. The different enzymes are still being occupied. So no matter how much I add in, it is limited by what? Okay, the number of enzymes that are currently have a free active site. The number of enzymes that are present in the mixture or whatever. Okay, so this is how you try to rationalize step by step. Okay, and then same thing for the next one, which is for your enzyme concentration. Same thing. Okay, if you notice, try to rationalize it out, draw it out if you have to. Okay, at low enzyme concentration, because there's very little enzyme. So if I add more enzymes to the overwhelming number of substrate, the new enzyme can straight away find the substrate, and then there'll be a linear increase. Okay. However, if I add a lot of substrate, you'll start to drop because now every active site is being filled by a substrate. Okay, so you can see the graph will start to decrease. So you can see, right, when this is where your figure interpretation comes in. Okay, linear increase. If you see the graph goes like that, what is this? Increasing at an increasing rate. But then if you see like that, it's increasing at a decreasing rate. So how do you understand this? By the gradient of the graph. So initially it's straight like that, but you can see if you go by the gradient, it starts to go like that, to go down because the rate of reaction is decreasing. Okay. Okay, so last two concepts very quickly before we move on to our practice questions. Okay, temperature and pH. Where does this stem off? Remember just now, proteins, enzymes are proteins. So if you apply a lot of heat, okay, or if you apply chemicals, pH, okay, the protein will denature. Enzymes will denature because they are fundamentally proteins. Okay, so if you heat it up, okay, when it comes to temperature, okay, take note that we're not talking just about enzymes here. Okay, we're talking about substrates as well. Substrates will be affected by heat. Okay, so they'll increase the kinetic energy heat. Okay, when we're talking about heat, wow. Okay, imagine it's very hot. What do you tend to do? You tend to move around, like wow, pace around. It's getting very hot. You tend to fidget a lot. Okay, so the enzymes and substrate will tend to collide more. Okay, up to an optimum level. Okay, where subsequently past this optimum level, your proteins, your enzymes, they can't take the heat anymore. What happens? They'll denature. It's too hot for them. Okay, okay. And then same for pH. There is an optimum level where the chemical environment is very nice. The enzymes enjoy it. They function at their best. But the moment you tip over this pH, they denature. Because chemicals, their bonds can be broken. Chemicals can break the bonds formed between and it'll disrupt the unique 3D conformation. Okay? So that is where your enzymes will start to denature and then they lose the active site. Okay, the moment your structure is distorted, okay, what happens? Your active site is lost. Okay, they are very delicate folding, and then you imagine it's like imagine this the enzyme, it works very nicely, okay, and then you go and smash it. What happens? The active site is gone, it's distorted. Okay, which is why proteins and enzymes are very delicate and why temperature and pH will affect them. Okay, okay. So I did a very super quick run through. I hope you can keep up. Okay, we're moving on to the practice examples now. So I'll be going through some practice questions to show you how to answer the question. And then there'll be a quiz at the end. Okay, so you can see how much you learned today. Okay, okay. So I just prepared 3 very short questions. Okay, so to more of consolidate your learning. Okay, but what I want you to learn from here, okay, it's not so much of, oh, I want to answer this question, okay? Yes, you can test yourself, but I think I want you to follow my approach, see how I approach the question and the answering technique, okay? Okay, so let's start. For cell biology, first thing to do, for me, right, when I did my A-levels, right, okay, or any question, any exam in particular, always make sure you read the question fully. This is to eradicate any potential careless mistakes, really understand what the question is asking for, identify which topic the question is based on, and then pick up keywords. Okay? So if it's a small insects which feed directly on phloem set in plants, okay, the salivary glands of aphids have secretory cells that make and release a variety of proteins that assist in feeding. Straight away, what comes into your mind? Cell biology, protein synthesis. Okay, and then with protein synthesis, think about it. Just now, what did I mention? Oh, where is protein synthesis occurring? What is needed for protein synthesis? Oh, okay, you should be thinking about the Golgi apparatus straight away. Okay, so you should think about chemical modification, stuff like that. You just think of like you should pop into your head. Okay, now the figure is a transmission electron micrograph of a small area of salivary gland cell of an aphid. Nope. What did I say? They just nice mention. Okay, so they want you to focus on Golgi bodies and mitochondria. You should immediately ask yourself, function, what is the function? What's the function? Okay, let's look at the question. Describe the role of Golgi bodies in the production of proteins in secretory cells, such as the salivary gland cells of aphids. There you go. This is what kind of question? Okay, this is a recall question. Okay, so recall questions, I think a lot of you may like it. It's just vomiting, regurgitation of your information, okay? Because it's just, if you memorize, you got it. Okay, if you didn't memorize, okay, try to take it step by step, okay? Calm down, most often you may be because you're very stressed and then you tend to forget, okay? But ask yourself, mitochondria, okay, what is it related to? Mitochondria is related to, okay, ATP production, okay? Golgi bodies, what is it related to? Okay, chemical, I remember just now what are the keywords? chemical modification, or glycosylation, phosphorylation, temporary storage, packaging. Okay, slowly try and jog your memory. Okay, so describe the role of Golgi bodies in the production of proteins. Okay, so what did I mention just now? The role of Golgi bodies are for the chemical modification of proteins. Okay, but do not stop there. Okay, notice that this question is how many marks? Okay, always judge by the number of marks. Okay, so 3 marks most often means 3 points. Okay, but doesn't mean you give 3 points and you're done. Okay, what you should be doing most often is that if there are 4, 5 points, okay, if you are try to give one more. Okay, so it's possible that for certain questions, right? Okay, they may, if 1 point may not necessarily be a keyword, okay, your subsequent point may back you up and get through that point. Because let's say maybe this point is not really as significant. Okay, but thank God you included the subsequent point. Okay, so here what we have, Golgi, chemical modification, keyword should be addition of sugars, glycosylation. Okay, what else did we mention? Packaging, okay, temporary packaging and sorting. Okay, packaging into what? Okay, into vesicles. And then what are we talking about here? If you go back to the question, notice what did they say? Okay, secretory cell. This is a secretory cell. This is secreting proteins, secreting whatever, but it's secreting proteins here, okay? So what should it be packaging into? Secretory vesicles. Okay, secretory vesicles to be secreted. Okay, and then what happens with the vesicles? Okay, the vesicle. Okay, so see, first I start off with describing the function, the structure and function of the Golgi. Okay, not structure, just the function of the Golgi, the chemical modification and temporary packaging and storage. Okay, and then what next? Formation of secretory vesicles. And then notice the question. Describe the role of the Golgi bodies in the production of proteins in secretory cells. Okay, so after you form the secretory vesicle with the modified protein, what happens to them? Don't just stop there, complete the story. The secretory vesicle translocates to the cell plasma membrane or cell surface membrane, whereby the membrane of the vesicle will fuse with the cell plasma membrane to release the contents, the proteins or whatever, into the extracellular environment. Okay, notice how I told the story. Okay, to make sure you really fully score the marks. Okay, so explain why secretory cells have large numbers of mitochondria. Mitochondria is for what? This one is more of an application and recall question. First mark, what is the mitochondria for? Okay, you should straight away think ATP production. Okay, now why do we need ATP? What is going on here? Secretory cells, what do they do? Just now, what did we cover? In question 1, you mentioned that they secrete. The vesicles need to move, right? So when they need to move, if you want to move, what you need? Energy. Okay, so notice here, this is more of a slightly easy application question, but you're basing your answer off what you answered in part 1 as well. Okay? Okay, moving on. Okay, mitochondria are partly controlled by the nucleus, but can also function independently. Suggest two features of mitochondria that allow them to function independently of the nucleus. Remember just like I covered the endosymbiont theory? This is what I mean by they can test your application. Okay, they didn't mention, oh, describe the endosymbiont theory here, but you notice that that's what they're asking for. Okay, you tap onto knowledge. Why do people say that the mitochondria are kind of independent? Because they are similar to prokaryotic cells. Why? This is where your feature should come out. So we can talk about they have their own circular DNA. They can divide by binary fission. Okay, stuff like that. Okay, so notice this is what I mean by you need to be careful when you're interpreting questions. Okay? Okay, very quickly moving on. So this is actually I think it's I'm not sure if this is a exam question before. Okay, so biomolecules. Okay, here we have a glycogen granule. Okay, now interpreting figures. My lesson, why I chose to give you all this question is, if you notice here, right, you see this, right? What is the first thing you should already do? Okay, representation of a glycogen granule. Glycogen granules are made up of glycogen molecules surrounding a central core. State the number of glycogen molecules shown in figure 1.1. It's very tricky. Okay, why? A lot of students, they see this, what do? They'll panic. They're like, okay, some students will see, oh my God, so many, how to count? But you think about it, do you hear what I just said? So many, how to count? Glycogen is a what? It's a polysaccharide, correct? It can't be there's so many here, what? It can't be this is glycogen, okay? So it's to scare you to waste some time now, okay? So ask yourself next, glycogen, okay, it's a polysaccharide. What is it made up of? Monosaccharides, right? Okay, so what is the monosaccharide? Glucose, correct? Okay, now, some students, they may get tricked. They see this thing. Oh, a glycogen granule, or one big circle means one. Okay, if you answer one, you got tricked. I'm sorry to say, okay? Why? Look at the diagram carefully. Okay, this, okay, is one glycogen granule, okay, but how many glycogen molecules are there? It's a different question. Glycogen is what? It's saccharide, right? Polysaccharide, okay? But look at this structure in the middle. These are helices, okay? When you're looking at this kind of illustration, right, this most often refer to proteins, okay? Which is why this is the tricky part, okay? And if you notice, if you erase this out, notice that the the glucose molecules are actually separated, okay? They're separated. This is not bound together. This is not bound together. There are gaps here, okay? So the answer should actually be 2. The number of glycogen molecules shown in figure 1.1 is 2. It's not 1. Okay? Now, name the type of molecule forming the central core of the glycogen granule. See, I answered it just now already. When you look at this structure, you notice the helices. What should they remind you of? Secondary structure of proteins. Naturally, when you see this kind of colored, okay, it won't be colored in your exam. So if you see this kind of illustration, most often it refers to protein structures, the secondary structure. So if you notice here, this is the alpha helices. So it's the protein. And then if you notice that there are actually sugars here, so I can, oops, let me show you here. Can you see this part? These are actually what I mean by glycoproteins. So these are the saccharide groups that are attached to the protein. Okay, so that one's just more for your information, but yeah, okay. So part 2, name the type molecule, protein, okay. Last, suggest the role of the molecule forming the central core of the glycogen granule. This is where it may get a bit confusing because you're just wondering what, okay, what is the protein's function here? Okay, just look at the diagram, trust yourself. Okay, what is it doing here? Notice that it's kind of holding the two glycogen granules together, right? It serves as the bridge, right? So what is the answer? The protein molecule is functioning as a bridge to bind the two glycogen molecules together. Why? Remember glycogen, what is the function? Has to be compact, correct? So here, it can be to hold the two glycogen molecules together, okay? Okay, so I hope that's clear, okay? Let me scroll down. Okay, B and C. So B first, explain how the structure of glycogen is related to its role in living organisms. I won't go through this. This one, as I mentioned just now, always go by your structure in relation to function. It's compact, okay? It's insoluble. It's what? Small, okay? What is the function? It's compact, so it can store what? What is it? It's a storage molecule for so forth, okay, in animals, okay? Now, describe how the structure of cellulose is different from structure of glycogen. Once again, comparison question. If you did your revision in terms of the tables, this should come as a breeze, okay? How the structure? Structure, we talk about what? When we are talking about formation structure, we're talking about bonds. Okay, so cellulose, what kind of bonds do we have for cellulose? Beta 1,4, glycosidic bond. Okay, and then what do we have for glycogen? Okay, so forth. How the structure of cellulose is different from the structure of glycogen. Okay, so from here, right, they're asking cellulose, how is it different from glycogen? So you should be answering from the perspective of cellulose. Okay, okay, so I hope that is clear. Okay, okay, so last question. I included here, I'm not going to go through the whole thing. I just want to show you how to approach this, okay? So the first question I show you was more to recall your, to show you how recall questions can be. Second question I wanted to show you is more of how to interpret figures and sometimes how figures can trick you. Now, this question, why did I include here, is more to show you how to approach essay questions. Okay, explain how pH affects the rate of an enzyme catalyzed reaction. Okay, Firstly, you're going to be you see these questions, I think some of you will say, because basically what you have to do is to really vomit this whole thing out, okay? Yes, you have to do that, but always remember 8 marks. Okay, the marks are here to guide you, okay? Essay questions, right? Don't be afraid to be more illustrative. So what do I mean by that? If it permits, right? If you feel it's appropriate, you are able and allowed to draw diagrams to help explain your answer. So here, pH, rate of enzyme catalyzed reaction. So automatically, what should come to your head? Optimum pH, denaturing, okay, increase, and then so you can see how do I illustrate all this without words? What is the key diagram? Okay, when you're studying pH, right? I'm pretty sure most of your lecture notes have this graph. So what is a good thing that you can do to help you aid your answering of the question? You can straight away start by drawing this graph. Okay, the graph of the rate of enzymatic reaction against pH. So you draw the graph. Okay, so now you have the guide. Okay, let me just draw a very... Okay. And then make sure to label it as well. Okay, so your rate of reaction, your pH, your rate of reaction, so forth. Okay, so this is marked as your optimum pH. And then you start your essay, okay, with explanation. So you go by a very sequential and progressive manner. Okay, you talk about, okay, at low pH, okay, so this is pH. Oh my God, sorry, my my writing is very horrible because I'm using mouse. Okay, so pH, okay, so start from low pH. As it progresses to higher pH, what happens? Okay, and then make sure to explain at the optimum pH what happens. This graph itself could net you 1 mark. Okay, and then you explain, okay, using proper sentences. Okay, at low pH, okay, rate of reaction is what? Is it low? Is it high? Okay, because we're talking about rate of an enzyme catalyzed reaction. So rate, is it high or low? Okay, next, as the pH increases, okay, what happens? Okay, does the rate of reaction increase? At the optimum pH, it's at its maximum. Okay, and then past the optimum pH, why does rate of reaction decrease? Oh, because the specific charge, okay, of the enzyme, the active site, the structure is being denatured. Proper sentences, hit all the keywords, and you will get your 8 marks. Okay, so yes, this is more of a mix of a recall question, okay, but you also need to take note that your marks and your diagrams, your illustrations can really help you go a long way, okay, because sometimes when biology, you memorize so many things, right, okay, you may tend to forget bits and pieces here and there, which is why diagrams really help to aid your answer and help you structure and be more confident in telling the story. So that is the thing about most essay questions, okay? If you're allowed to draw diagram, or if you have to draw the diagram, right, okay, really just draw the diagram to help aid your answer, okay? So, and most of the time, even if the diagram, they won't give you marks. It's not like, unless you can draw it wrongly, it's not like they will penalize you for it. Okay, so your diagram is more of like, can be for working, but it also can be part of your answer to help you guide your answering of the essay, or your writing of the essay. Okay. Okay, now, that was a very quick, super speed run. Okay, so I hope everyone is still alive and keeping along with me. Okay, so for the next part, okay, there's actually a quiz to help you. Don't worry, this one is MCQ quiz to help you test your understanding. So it's a Kahoot quiz. So I'm going to share my screen. Okay, and then okay, so you can test your revision. Okay. Okay, so everyone, you can log in on your phone or your, I don't know, your computer, and then we can proceed with the Kahoot quiz. So there's only 11 of you here. So, okay. So this is the last segment before we end off, okay? So the quizzes are 3 questions for each of the topics I covered today. So they really help to test your understanding, okay? They're MCQ questions, don't worry, okay? Okay, nice. Okay, any more people? Okay, very cute emoji, nice, okay. Hey, this one not chemistry, 1, 2, 3, 4, 5, 6, 7, 8. Okay. There's 3 more. Okay, 2 more. Okay. There's still 2 more. Okay. Okay, never mind. I think I shall just start. Okay. Okay, I'm just gonna start. Okay, everyone, I hope you're ready. Okay. Okay, all the best, guys. Very simple questions. Okay. Which description applies to cellulose? Okay, so just really be careful. Okay, I see most of you got it correct. Okay, 1A and D. Okay, so you might have gotten tricked. Okay, so biology questions, they can be very tricky. Yeah. So later, I'll quickly run through some MCQ tips for you all. Okay, before we end off. Okay. Congratulations, JM. Okay. Okay, next question. Which term most accurately describes catalase, collagen, and hemoglobin? So what are catalase, collagen, and hemoglobin? Okay, so this is where it gets a bit tricky, okay, so I purposely put in polypeptides rather than proteins, okay, because catalase, collagen, and hemoglobin, they are all proteins, polypeptides, but collagen is not for those who chose globular proteins, collagen is not a globular protein. You got tricked. Catalase, okay, it's an enzyme, but collagen and hemoglobin, they are not enzymes. Okay, remember that. Hemoglobin, it may sound like an enzyme because it carries out a function, but it's not. Okay, so be very careful. And collagen is a structural protein. Okay, so this is what I mean by bio questions can be very tricky. Okay, next question. Okay, I don't know what face is that. Okay, but congrats. Okay, next. Okay, so this is a tough question, I think. Okay, very good, very good. Okay, I actually thought more people would get this wrong. Okay, so just to go through, X breaks all bonds in the protein molecule except covalent bonds. Now, what covalent bonds are present in protein molecules? Your peptide bonds and your disulfide bonds. So that means your hydrogen bonds, ionic bonds, and hydrophobic interactions are all broken. And that will mean your quaternary is broken, your tertiary is broken, your secondary is broken. And that leaves your primary, which are bounded by peptide bonds, and those are covalent bonds. They are not broken. Okay, so just to go through. Okay, okay, nice. I see the scoreboard fluctuating a bit. Okay, next question. Okay, so this is why I think this is why just I write 5 microliters. Okay, really take your time to understand the diagram. Slowly, slowly. Okay, very good. Okay, so I can see one of you chose. Okay, I think you guys got tricked possibly by the cell surface membrane. Okay, so be really careful. Really see cell surface membrane. They are most often labeled, right? You can tell by just a single line. When you see it's a thicker line, right? Okay, that tends to be the cell wall for plant cells. Okay, cell surface membrane most often they're illustrated by a single line. Okay, in the electron micrographs. Oh wow. Okay. Answer strict. Okay. Okay, so this is more of a sequential. Okay, really try to understand the sequence of events. Okay, I think this is a bit tougher, I see. Okay, so when it comes to sequential questions like this, right? Okay, wait, let me see. Most of you guys chose 3. Sorry, the red one. 1, 4, 3, 2. 1, 4, 3, 2. Okay, I see why. Okay, so when you're answering this kind of sequence questions, right? Okay, if it's in case you don't know how it starts or how it ends, always try to pick one end first. So if you notice, mucus is secreted from a goblet cell in the trachea. So when you look at all these 4 options, right, what is the final step? It should be the secretion itself. So 3 should automatically be at your last. So you should eliminate 1 and 2 already. So that leaves blue and green. Now, then you go to the first step. So the difference is separation of a vesicle or addition of the carbohydrate. Now, if you recall for the Golgi, what it has to do first, it has to form the thing first, right? Before it releases the vesicle containing the modified protein. Okay, and then you check your middle steps, are they correct? Okay, so that's the trick. Okay, wow, okay. The last few questions. So keywords are, for this question, hydrolytic enzyme activity. So what organelle has hydrolytic enzymes? Okay. This is where you should recall each organelle's function. Okay. Lysosome is for what? Mitochondria is for what? Nuclei is for what? Okay, very good. Okay, so those who chose A and B, okay, I think you may have gotten tricked. Okay, just try to remember, go through each of the organelles and their functions a bit more carefully. Okay. Okay, nice, Yiting, you're catching up. Okay, few more questions and we are done. Okay, temperature X. What is happening here? Keywords, temperature, what was the keyword I kept using? Okay, very good. Okay, so yes, I know one of you got tricked by the kinetic energy. Okay, but recall for temperature, it's more of everything has high kinetic energy. So when they collide, they form maximum number of enzyme substrate complexes. Okay, wow. Okay, last 2 questions. Okay, I actually mentioned the answer for this just now. Okay, well done. Okay, tertiary. So be careful. I mentioned there are specific, and the 1% for quaternary, there are certain enzymes that function in quaternary structure because they need multiple polypeptide chains together. And there are some that are just on its own. So tertiary and quaternary. Okay. Okay. Wow. Tough competition. Okay, last question. Oh, okay. So this was more of a bonus question because I didn't go through inhibitor. So whoever can answer this, yeah, good revision. This is more of a bonus question. Okay, well done. Okay, so this one I didn't really go through and there's not much time, so I can't really. Okay, but I'll try and see. So yes, A is more of the most accurate description. Competitive inhibitors, actually just be careful, they do not have the exact same shape, but they tend to have more of a very similar shape. Okay, they are structurally similar, not structurally exactly the same. Okay, and non-competitive inhibitors cannot have any shape. They must have a shape specific to the allosteric site. Okay, which is where they'll bind to. Just that they don't bind to the active site, they bind to an allosteric site. Okay, so congratulations. Okay, so unfortunately this one, yay, okay, congrats. ZX, yay. And then last one is the emoji person, right? Okay, so yikes. Okay, so I know for those of you familiar with Walmart, there's actually a giveaway, but unfortunately, because for mine, I'm not done with my curated notes yet. So yeah, I'm sorry, but congrats. This means your revision was good. Okay, so before you guys leave, let me just round off with. Okay, let me just round up today's lesson. Okay. Okay, so before you guys leave, let me just do some final ending up stuff today. Okay, so today's content, right? They're all from Overmark's curated notes. So for biology, because I'm recently joined, okay, I'm still in the progress of making the bio ones. There will be all three booklets will be out by August. The first one, which will be out by June. Okay, so if you want to order for the other subjects, I've listed the QR code here. So the notes you can see today are actually, as I said, snippets from the curated notes. They'll contain the key concept, they'll contain examples, really everything you need to revise. So it's like everything compiled together, okay? So if you need more help, okay, so for tuition itself, currently I'm not doing any one-to-one or group tuition because I have other commitments. But in the future, actually, if you want to discuss more, if you need some side help or anything, or if you want to discuss anything about one-to-one or group tuition, feel free to contact me at these two, my number or my Telegram handle, Telegram preferred. Okay, now crash courses, right? Okay, so this month the crash courses are actually coming up in June. Okay, and for me, because I have the created notes for the first module done in June, I'm hosting one module for the crash course. It costs $200 for one module, which I'm offering. Sign up your friends to enjoy 10% off and first come first serve basis. So currently for bio, we only have one module. QR code is there. If you're interested, you can sign up. Okay. What are we doing at the crash course? It will be a 4-hour session. Won't be so rushed like today. But what I'll be doing is covering the key concepts of the different topics. I'll be covering commonly tested questions. Same mistake, very similar format today, but more expanded because I got more time to really help work with you guys, and you'll be more in person, so I get to help understand all your concerns and questions as well. Okay. So the location will actually be at Spatial Collective at Toa Payoh. So it's a very nice, I haven't actually been there, but I heard it's really nice. Okay, so the topics that we're covering, as I mentioned, because different schools teach at different paces. For me, what I'm going to cover is cell biology, biomolecules, enzymes. Now, this is where it may get a bit different. I know stem cells, some of you will cover later. So stem cells, respiration, photosynthesis, cell signaling, and homeostasis. Okay, so these topics are applicable to both J1s and J2s. So J2s, you probably should have covered all. J1s, it's more for cell biomolecules, enzymes, and then the rest is to help you prepare for your later part of the, you should be learning the rest of the topics in the later part of the year, okay? So these are the key topics. Price as listed just now, $200, okay? And before you guys leave, if you really enjoyed today's revision, because I'm new to Overmark, please leave a kind Google review, and or if you have any feedback on how I should improve, please feel free to leave the Google review. And as well, or if you have very personal feedback, you can feel free to Telegram me, or if you, as I mentioned just now, if you want to discuss anything one-to-one tuition or whatever, feel free to Telegram me. Okay, so I'll be sending out the some of the slides that I used today on the Telegram channel. And if not, I wish everyone good luck for your mid-year exams. And then yeah, looking forward to seeing you sign up for the crash course. Okay, so thanks everyone. Hope today was helpful and good luck studying. Okay, so okay, you guys can leave. Bye bye. Take care. Okay, thank you.